Good afternoon. My name is Praneeth Namburi, and I am a research scientist at the Institute of Medical Engineering and Sciences at MIT. And I also have an appointment at the MIT Nano Immersion Lab. Today, I'm going to talk to you about dance-inspired investigation of human locomotor coordination. So first, let's talk. Our intuition suggests that skilled dancers produce beautiful and coordinated movements. But what is coordination? Let's start with the dictionary definition. The dictionary offers two definitions depending on which dictionary you're looking at. Coordination is the organization of different elements of a complex body or activity to enable them to work together effectively. Or it's the ability to use different parts of the body together smoothly and efficiently. If we look at what is common across these two definitions, it is the idea of relationships between parts. If we look at what is different between these two definitions, it is the type of relationship. So today I'm mostly going to talk about how to identify and quantify relationships between different parts. In the context of movement, motor coordination is an orchestrated movement of multiple body parts to accomplish actions like walking. Coordination is applicable in our daily life, not just in the context of specialized movements. But the question is, how do we measure motor coordination? Traditional metrics, like the ones I have put up here, are based on angles, joint angles, positions, and the relationships between them and how they change over time. They also include derivatives of positions such as velocity and acceleration. And while these have offered excellent insights, the primary challenge with using such metrics is that it's not straightforward to compare coordination across movement types. In our case, for what I've talked about, it's walking and dancing. It could be any different movement. So what I'm interested in is if I walk and then do some jumping jacks, uh, it's hard to know whether I am more coordinated in my walk or my jumping jacks. There's no straightforward way of measuring the two. So since both walking and dancing are coordinated movements, we should be able to quantify the amount of coordination independent of the movement itself. So here I'm going to share a video clip of two dancers. So to solve this challenge that I just talked about, we're going to draw inspiration from dancing. Here you're going to see two dancers move around to the same music, but they are moving through different positions or postural pictures in space. I'm going to optimize for video clip. 1 second. All right. These two dancers are world champions in their field. They're Ricardo and Yulia. And despite how Ricardo and Yulia move differently, move their bodies differently, they are still dancing with each other and are dancing to the same music. Different movements, same rhythm. So we decided a good place to start is to observe how the dancer's body represents rhythm. And in this case, we are going to use motion capture. What you see in this picture are two dancers and a coach. In the background, you see dots, which are basically infrared reflective markers, which track specific points on each dancer's body. And I'm going to share this clip. And as the dancer moves, you see the points move in the background. I'll go into how we actually use this in a moment. And let me summarize our strategy first. So our strategy to quantify coordination is to monitor muscle length oscillations, because rhythm is represented in muscle length oscillations. We think that this can be used to quantify coordination. So today I'll talk about what this might look like. Now, intuitively, when we run, we listen to music and we might pick a specific cadence of our interest based on how we're feeling that day. So the idea of rhythm generalizes to more than just dance. So here we designed an experiment whereby we monitor length of muscles and rhythms in the muscles while on a treadmill listening to music. I'll play this clip again. So this is what the experiment looks like. Now, we are using each of these little black tapes, and the white dot in the middle is infrared reflective marker, and we use motion tracking cameras to track the position of each of these points in 3D. And an infrared picture is shown in the middle. And what I'm interested in is not the shape of the muscle itself, but the length of the muscle and how that evolves over time as I run. And here are two examples of how the shape is different, but we have to crunch the numbers to get the length. So here's an example of what the length looks like. On the x-axis below, if you look at the black line, is the position of my left heel. So every time my left heel goes back down in my running rhythm, it's 2 steps. And on the green line quantifies the length of this muscle system or a group of muscles called erector spinae in my back. And you can see that muscle length changes, sometimes more smoothly, sometimes less smoothly. And sometimes when I enjoy the music, the amplitude of these oscillations can increase and they can become smoother. The idea here is that each of the different segments are synchronizing with each other. So we could think about a less synchronous or coordinated state, or a more synchronous and coordinated state on the right. I'm going to show you what the running videos look like. So this is the less synchronous version of the erector spinae muscle, and here's the more synchronous version. If you just look at how the shape of the muscle and how these points are moving through space, there isn't an obvious visible difference, but in terms of the relationships between the points, there is a much more clear difference, as you could see in the green line. Now, synchrony or smoothness in one muscle system does not guarantee smoothness in other muscle systems. So here I'm showing you in green length of the erector spinae muscle, and in orange length of the latissimus dorsi muscle. Now, towards the left of this plot, the latissimus dorsi is arguably less coordinated, and on the right, these points are more synchronous with respect to each other. The amplitude increases, oscillations become more obvious and smoother. And in the green line, I'm showing the length of the erector spinae muscle, and there is no real discernible change between the two phases when one muscle changes state, so to speak. So now pay attention to both muscles in this video. All right. So but it is possible for both muscles to represent rhythm. Interestingly, here, if you this is yet a different segment from running, and if you compare the green and orange lines, the dominant oscillation frequency in the orange line, the lat, is about half that of the green line. So for every 2 oscillations of the erector spinae, which means step step, there is 1 oscillation of the lat, which is basically going for left and right. Okay, so finally, let's look at coordination across individuals. So far, what we talked about in the context of running is coordination or synchrony within one muscle and between different muscles. Now, we are going to look at coordination across individuals. In this case, we return to the example of 2 dancers where we are monitoring the length of the same muscle, the erector spinae. Show you what the setup looks like. Okay, so the coach is there to try to guide them through doing this same movement better. So this is before the coach guided through on the left. Let me optimize for video clip again. And on the right is after they took the lesson with the coach. So let's compare the length changes in the same muscle but in 2 different individuals. Now, the green line represents the muscle of the stationary partner. So in this figure, one person is spinning in and out, so that's the spinning partner, and the other partner is stationary, so that's the stationary partner. We are going to monitor the length or the separation between the dots that you see on the screen in the stationary partner and the spinning partner. Before the lesson, arguably there isn't a huge relationship between how the length of the muscle in blue for the spinning partner relates to how the body of the stationary partner responds. But on the right, in this example, you see as the blue line increases, the green line also increases, meaning as the spinning partner is storing elastic energy in her muscle, the stationary partner's body is also responding to changes in the stationary partner's body. Now, of course, this is one example, and we need to do more experiments to generalize to see if this generalizes across dance forms and figures. Now, so far I have talked about using motion capture to observe rhythms in elastic tissues. In the future, we are also planning to use ultrasound to look to quantify rhythms in the deeper lying tissues and also to reduce the interference with fat and skin moving along the muscle tissue. And I'll show you this clip. As I move up and down, you should be able to see the muscles contract and relax using ultrasound. So we are hoping to quantify rhythms using video processing. So to summarize what I've told you so far, I've tried to argue that synchrony in muscle length oscillations is a relatively unexplored metric for coordination. And today I talked about how we could use this metric to observe changes in a state of coordination within a muscle, between muscles in the same individual, and across individuals, in this example, dance partnership. And this metric hopefully can generalize to any movement discipline. I would like to thank the 2 PIs I worked with, Luca Daniel and Brian Anthony. Armin is the dance coach who is instrumental in this project, and below are staff and Europe students who also help with the project. I worked with Miha on the ultrasound part, and will be developing this aspect in the future. And I'd like to thank our funding agency, NCSoft, and I'd like to thank the MIT Nano Immersion Lab.